Right, good afternoon again, everybody. I think we're ready to start with the second presentation, or the second half of this afternoon. I'm sorry there was a hiccup at the start of the first one when I welcomed everybody back. What I was about to say was how good the first 2 days had been, and I was looking forward to this afternoon continuing that excellent um, explanation and talk about what's been going on with COVID, and that certainly happened with our first presentation this afternoon. Before we start, just a word of thanks to the organisers, to the BSMT for putting all this together. It really has been an excellent few days, and will continue tomorrow. I'm always grateful for the updates that I get from these conferences, especially now that I'm a few years retired. It's a really good way of catching up on so many aspects. Now, as your screens have shown, we must also thank the sponsors for this meeting. Without their help, without their support, without their financial input, none of this would be possible. And I think we've all benefited enormously from that support that's enabled this conference to take place in this way. Next talk was the second one for this afternoon. Again, is very much focused on genomics work. This time going back to the main topic of SARS-CoV-2 itself, the virus, and what's happening to it. And very pleased that we've got Professor Nick Loman with us to talk about this. Who's Professor of Microbial Genomics and Bioinformatics in the University of Birmingham. I'm sure he's spending all his time at the moment working on COVID and the variants, and feeding into all the work on sequencing. He's been doing this sort of work for a good number of years now, and has spoken to BSMT on previous occasions. Now, of course, with the Indian variant being one of the headline news items this lunchtime, just before we started this conference session today, it could not be more topical. So I'm hoping now that we can just switch straight into Professor Nick Loman's.
Hi there, and thanks very much for the invitation to speak today. So I'm going to talk about coronavirus genomics, and I'm going to focus on how we've managed to set up coronavirus genomic surveillance in the UK, and then think a little bit about where we're going to go with that in the future, both in the UK and worldwide. So let's start at the beginning. A cluster of fatal cases of a respiratory disease were detected around Christmas time 2019, with a focus around the Wuhan seafood market. And while it's not entirely clear whether the virus spilled over directly at that market, or whether this was the first super spreading event in the pandemic, of which there have been many subsequent, that's not clear. But we do know this is the first time the world was alerted to a new pandemic agent. And whilst speculation as to the nature of that agent was rife, it was revealed in early January that we were dealing with a new coronavirus. And that coronavirus took a little while to be named properly, but was eventually named SARS-CoV-2. And we really do have this person to thank. This is Prof. Zhang from Fudan University. And he took a pretty bold move in early January by agreeing to release into the public domain for the first time a genome sequence of this new viral agent, which turned out to be similar but still quite distantly related to other coronaviruses, including SARS-1 and others like MERS and other endemic coronaviruses. And this was a brave move because he really circumvented some of the formal processes in China for releasing data, recognizing the urgency of the situation. He was recently featured in the Time 100 People of the Year in 2020, and I really do think Prof. Zhang is a hero of this pandemic, because this really kind of points out how important genomics is in the response to a new outbreak or epidemic virus. The genome gives us a really good idea about the biology of a pathogen and its likely features. Critically, we know that we need a genome sequence to be able to develop PCR-based tests, the mainstay of diagnostic testing globally. And there is clearly a link to vaccine development, either indirectly by knowing the protein annotations and understanding the sequence of the spike protein, or actually directly in the case of the new, very successful recombinant viruses using virus vectors, and of course the incredibly exciting development of messenger RNA vaccines. There's an absolute direct link between having genome sequences and being able to produce those vaccines. So days matter in a pandemic to be able to get a genome sequence out. And this also represents a triumph of the idea of metagenomic discovery. As you know, previously, before these techniques were available, isolation of novel viruses typically depended on growth in some kind of tissue culture, identification often through microscopy, and the genome sequence would follow quite a long time after that, in the old days, potentially years. Now we can use metagenomics as a one-shot assay to detect a novel pathogen and to sequence it entirely. And that can be done, as was done in China, and by several groups very quickly. So that represents a kind of step change in our laboratory capability. Interestingly, we don't really have that capability routinely in clinical diagnostic labs in the UK yet, but it's certainly coming, as I think the previous speaker alluded to. Okay, so we have a genome around the 10th of January, and we got involved. Like so many groups, we pivoted. We didn't pivot that far because in Birmingham we already work on epidemic viruses and have been tracking with genomics epidemics, including Ebola virus disease in West Africa, Zika, yellow fever, and others, and had a ready-to-go project called Arctic that was really all about tooling up labs in order to be able to sequence outbreak and epidemic viruses when they hit. We didn't really anticipate that during the duration of this grant there would be such a huge pandemic virus emerge, but of course it made perfect sense for us to pivot very, very quickly to looking at coronavirus. And Josh Quick, who is a UKRI Future Leaders Fellow at Birmingham and works as part of the Arctic network, was able to very, very quickly put together, using that original Zhang genome, a bespoke sequencing protocol for coronavirus. And the way we do that is we use a multiplex PCR tiling approach. So we divide the genome up into around 100 fragments, each fragment about 300 bases in length, and the genome's 30,000 bases in total. And we cover that genome by that amplification scheme. By sequencing those PCR products, we can reconstruct most of the genome quite effectively. And the reason this approach is particularly nice is that, unlike a metagenomics approach, it's on target. So pretty much all the sequence reads you get come from the virus itself. So there's no wasted sequence, and that means we can have quite a cheap assay that is also able to function even down to quite low viral copy numbers, so up to CTs of around 30 or so. And that means we can get good results for a large proportion of the cases that are reported. So in common with our with everything in this project, this is all open source, and because we were able to develop that very quickly and release it open source, it has been very, very widely adopted globally. It's hard to estimate exactly how many sequences have been generated with this protocol, but conservatively at least 50%, and probably a great deal more have used this protocol, which is incredibly gratifying. We've also been sending out aliquots of our primers to labs globally to help them on their way, which has been a great success. In the UK, of course, we have a fantastic genomics sequencing capability, and we wanted to leverage that network effect in the UK through the Coronavirus Genomics Consortium that's led by Sharon Peacock at the University of Cambridge. And this is very much a distributed effort. We have around 16, 17 universities contributing sequencing, partnered up with their local diagnostic sequencing laboratories in the NHS and public health agencies. We also have the benefit of the Sanger Institute, who are partnered up with the large lighthouse testing laboratories. And we were able to stand up, using those techniques that I mentioned, and using the goodwill and enthusiasm of many, many researchers, hundreds of researchers across the UK, stand up a sequencing network very quickly in late March, as it was apparent the UK was undergoing a large wave of coronavirus cases. It's kind of a complex system, but just to show you in schematic form how integrated it is, and I'll talk about some of those pieces, but the important thing here is that sequence data and metadata all flow into a central analysis resource and flow out again to the public health agencies, back to the NHS laboratories, and also critically to open access databases. And this has been a jewel in the crown of the UK the UK response, to have really so much open data available. In Birmingham, we are just one node, but working with a relatively small team of postdocs in my lab and with diagnostic folk from QE Hospital, Heartlands Hospital and other hospitals in the West Midlands. We've attempted to sequence almost 7,000 samples at this point, primarily on the Nanopore platform. We are able then to sequence quite quickly. Our turnaround time can be down to as little as 24 hours if required, more often a few days. And we are able to report back directly to hospitals what lineages they have, variants of concern, and critically and interestingly for us, trees that can be used for molecular epidemiology purposes. How do cases in the hospital relate to each other? Nosocomial transmission has been a big aspect in this outbreak. So how do cases relate to each other? And rule in and rule out outbreaks, and that's been of great use to the infection control team and health protection teams looking in the community. So 7,000 genomes in Birmingham, making up a total of nearly 470,000 genomes across the UK. And as I say, the UK has been the kind of pioneer of your open data sharing. It's also been the majority contributor of genomes worldwide for quite some time. At one point, over 50% of all genomes worldwide were produced in the UK. Now, as the cases come down, that number is more like a third. And in Birmingham, we're extremely fortunate to be able to host a node of the cloud infrastructure for microbial bioinformatics. That is where the data is processed from the sequencing laboratories and integrated, and we rely heavily on Sam and Radislav, pictured here, to keep these servers running and to keep the software running that allows all of this data to be processed. And the way this works is it's processed on a daily basis. New genomes flow in from our sequencing centres. They're integrated with metadata. We generate, we do quality control and generate outputs like lineages, trees, and other products that can be pushed back to the sites and back to the public health agencies. And this is a really unique feature in the UK that I don't think has been replicated elsewhere. So, okay, so we've generated a lot of genome sequences, but does it, what does any of it mean? How is any of it useful? Well, the first, I think, very, impressive demonstration of genomics was sadly retrospective in nature, but nevertheless generated quite useful information for policy making purposes to understand the role of travel. And the question we wanted to ask in early summer last year was, how did SARS-CoV-2 actually arrive in the UK and spread, and what's the kind of characteristics of that spread? And then to pose the question, and obviously this is an ironic question in the circumstances, but how could genomics help prevent a second wave? We clearly didn't prevent a second wave, but we certainly predicted a second wave with the emergence of the Kent variant, of which more later. We're also obviously interested to know over that course of time how new mutations in the virus might impact characteristics of the virus, including transmissibility and antigenicity and immune escape. So you actually answering the question of how did SARS-CoV-2 enter the UK is actually quite difficult, and it was particularly difficult back in spring 2020 for two reasons. Firstly, this is a recently emerged virus, so when we look at phylogenetic trees and we predict the most recent common ancestor of all cases circulating in the UK, we can date with really quite precise accuracy, leveraging the fact that the virus evolves at a constant rate of around 2 mutations per month. We can estimate that the emergence of that outbreak was around the start of December 2019, and in fact that actually correlates beautifully with the first officially recorded case in Wuhan in China. Now the difficulty comes that because a very small amount of time has elapsed, and time is directly related to genetic diversity, it's only through time do we generate large numbers of mutations. It's not a function of how large the epidemic is, it's a function of time. So what this means is that when we look at a tree, particularly back in springtime last year, there is not much diversity in the tree. And although this tree on the left here looks quite like it's got a reasonable amount of diversity in it, and you can certainly see it's divided into multiple lineages or clades. If we compare this to something like HIV, which has been circulating in humans for over 100 years, we have, SARS-CoV-2 makes a tiny part of the diversity that we would see in HIV. It's incredibly clonal compared to a well-established pathogen, and this makes genetic inferences difficult, because basically what this means is when you have imports from other countries, a lot of the cases look basically indistinguishable, or at least very similar. The second problem we have is certainly back then there was limited genomic surveillance, and most countries had no genomes or very few genomes, and so it's hard to identify a source of any particular lineage. But Ollie Pybus in Oxford, working with the COG consortium, did attempt to do this using some clever methods, and the sources of data he uses here is the concept of the time to most recent common ancestor. So effectively divide up the outbreak into lineages, and each lineage that has a non-UK genome at the base and then a set of UK genomes is considered to be a separate UK transmission chain. And this has to factor in certain lags, the lag for the patient to be input, to come to the UK, and then the lag for that case to be detected, and then we might not detect the first case, we might detect the second or third. So there are certain lags in this process that needs to be factored into an analysis. But what you could tell, which is pretty interesting already, is that for each of these different lineages, of which there are many, most of them actually do not result in a great deal of downstream transmission. They are imported and they die out. maybe result in less than 10 cases. Some generate larger clusters in the order of hundreds, and a few go supernova and result in thousands of downstream transmissions. And this is seen across the world. And this is important in understanding SARS-CoV-2 because this is a controllable virus. If you can stop those large lineages from transmitting, then you don't have a huge R. And we know that the R is very, very heterogeneous, has this high dispersal factor. So most lineages don't do much, and some lineages go off and do really well. And so that's an important finding. So where did these cases come from? So it's quite difficult to understand that from the genome data alone because we don't have good sampling from other countries. So the way around this is to incorporate travel data. So Ollie estimates what we call the importation density by matching up travel data. How many travellers are coming to the UK over this time period, of this critical time period of spring, it's winter spring 2020, and where are they coming from travel data? And in that case, you get a metric which is the estimated importation intensity. And when we plot that estimated importation intensity, we get this black line here. What we can see is that we have increasing intensity of imports of SARS-CoV-2 worldwide, right up until around March 15th, when we started to introduce lockdown type measures. And what you can also see is from the histogram of lineage TMRCAs, you can see that these lines actually fit quite nicely together, factoring in that lag that I mentioned. So with the lag, the detection lag, actually these curves overlap pretty nicely. So we finish this off by overlaying the travel data in terms of port of origin, and we can say where we estimate these cases come from. This is not definitive, but we can make a pretty good estimate that for the over 1,300 separate lineages, separate introductions that we detected into the UK, we can guesstimate where they came from. And we see that although around February time, you will have remembered we had signs up warning people that they, if they'd recently come from Wuhan, they should be self-isolating. In fact, most of our imports came from the continent, from countries like Italy, France and Spain, and actually very, very few, except for a few cases early on in late January, almost nothing came directly from China. So our border policy was wrong, and clearly we imported a lot of cases from places where there was a high density of coronavirus and high mobility to the UK, so exactly the countries you'd expect in that case. Sadly, we repeated exactly this pattern again, despite we had written this up and it was communicated, but we repeated exactly the same pattern, you'll remember, in late August, September, where we imported very large numbers of cases of a variant with the mutation A222V from Spain. So, so we have now a feel for how coronavirus behaves and how we can use genomics. And actually our life became easier in the sense that as time goes on, more diversity appears and more genomic surveillance goes on. It becomes easier to do this kind of importation work. And we switched gears now a little bit to looking at mutations and functional mutations. And over the period of 2020, functionally the virus did not seem to change a great deal. It seemed fairly uninteresting from the point of view of large phenotypic changes. Probably the most notable phenotypic change for most of 2020 was associated with a single mutation called D614G. That mutation, which was in the receptor binding domain of the spike protein, the receptor binding domain being important for being the part of the spike protein that binds to the ACE2 receptor in humans to aid entry into human cells, had been changed, and the prediction was that this would increase affinity for the virus to the ACE2, and therefore potentially increase transmissibility. And this is a good example of a kind of selective sweep of a particular mutation that was thought to confer increased transmissibility. And so, although the wild type virus had this D form, this G form emerged very, very early on in late January in China, actually, and very, very quickly through February and March swept to fixation and became the dominant form. Just a few examples of the D form exist still globally. They are out there, actually, interestingly, in Africa. But other than that, the virus remained uninteresting kind of from a functional evolutionary point of view, and was mainly the genome data was used for molecular epidemiology. Up until late November, December time, when we noticed that Kent was undergoing a surge of cases. And Kent was notable for being a region that was in tier 3 restrictions, unlike London, it was in tier 2 restrictions. Like Birmingham, they were higher tier, and suddenly the county of Kent seemed to be undergoing a large surge in cases, and so the question was why. And this story is very nicely depicted, albeit with some quite silly photos of myself and colleagues in the Guardian. So I do recommend that article. But they paraphrase me saying what we were trying to work out in collaboration with PHE is what was going on in Kent, and what epidemiology would explain the cases in Kent surging, but not elsewhere in the country. And we joked that had everyone in Kent secretly gone to an illegal rave, there wasn't a clear epidemiological explanation, in fact, and this became a puzzle. So enter genomics, and I remember vividly a meeting where we brought together some epidemiology with the genomics in early December, and said, well, what's going on in Kent? And we looked at the trees. There's big trees that we generate in the COG consortium, and we saw that that very, very strikingly, over half the cases in Kent were belonging to a new lineage, which was named B117, although everyone still calls it the Kent variant. And the Kent variant is now known as the Grinch that stole Christmas, because it heavily influenced government thinking around Christmas regulations and lockdowns. But the fact that a particular lineage was surging like this was very, very striking, because when we'd looked at previous surges in other cities, like in Birmingham, surges were typically associated with multiple lineages, with large amounts of genetic diversity, and surges were almost always put down to epidemiological factors, relaxing of lockdown measures, and behaviour, and just the virus spreading and exploiting more opportunities to spread. In this case, it looked like the lineage was possibly driving the spread for the first time. And another crucial piece of information that we had was that Andrew Rambo had recently attended a meeting of WHO to discuss the surge of cases in South Africa caused by their very own variant, which is termed B1351. In that case, a mutation was noted in the South African strain called N501Y, also called Nelly, and Andrew had been looking independently for examples of Nelly in the COG dataset, and he'd found this lineage B117 as containing that 501Y mutation. So suddenly we felt that for the first time a change in the virus might be driving a difference here. When we actually look at the genomics and look at that lineage in a bit more detail, we were quite taken aback, and that was for a couple of reasons. One was that this virus had a great deal more mutations associated with it than we were expecting to see. So rather than the 2 mutations a month rate, it seemed like around late September, the virus had gone through a burst of accelerated evolution and accumulated around 20 more mutations than we would have expected for that period of time. Again, after September, it then seemed to go back to its normal clock rate, as illustrated on the right here. Even more notably, those extra mutations that we see seem to be functional in nature. So a large number of them were non-synonymous changes in the spike protein, including that N501Y, including including deletions in the spike protein that would be likely to affect the N-terminal domain of the spike, and also a notable mutation that we think is getting more and more important, which is 681H, which is in the furin cleavage site. So quite a lot going on in this lineage that we hadn't seen before, and also that look notable. And we think that explains the results of the modelling that proceeded, which was basically to try and estimate transmissibility. And the modelers, by looking at genomics and by looking at the diagnostic S-gene assay, which was also a marker for this strain in the thermotacpath assay, were noting a huge advantage. Every time this particular variant ended up in a new part of the country, it outcompeted what was there already, the wild type variants, if you like. It just did seem to do extremely well. And the estimates early on were 70% more transmissible from this data, although that's been refined down a bit to more like 50%. But at this point, and this has been replicated worldwide, everyone is convinced that this strain was significantly more transmissible than the wild type. It also has some signs of increased mortality, although it doesn't actually seem to change mortality rate for admitted patients. It does seem to result in more patients going to hospital and therefore more deaths. And you can track all this through a website called MicroReact. There are other dashboards available now, including Next Strain and Outbreak.info. But you can see here strikingly how B117 in green here came to dominate the UK and eventually worldwide, just like that D614G did early on. And other countries are on the lookout for variants as well. A notable one that we were involved with was the discovery of the P1 variant in the Amazonas region of Brazil. And I don't have much time to talk about this, but the P1 variant is notable because there is a suspicion. It also includes that N501Y, but it also has several mutations, 417 and 484K, that are thought to significantly change the shape of the spike protein and therefore have an effect on antigenicity and potential immune escape. And the reason people were concerned with immune escape in Amazonas was that they had such a huge first wave of coronavirus that it was estimated they may have hit a herd immunity threshold early on in the pandemic. And then they had a second wave, even larger. And so the question was, could P1 both be more transmissible than wild type, but also result in reinfections? And so that's not entirely clear at this point, but it's what we need to watch out for. And this is really the kind of summing up of the talk, is that we are really now in the game, I suppose, of variant discovery and variant surveillance with genomics. And what we really worry about are those two aspects. Transmissibility, the virus will always be selected for greater transmissibility if it's possible, and immune escape, a big change to antigens would have impacts in terms of reinfection and in terms of how well the vaccines are going to work. Now, most of the data at the moment around vaccines is reassuring, although some strains, particularly B1351 from South Africa, has some evidence of reduced susceptibility to neutralizing antibodies. The expectation is that the vaccines will currently work against all the circulating variants. But what we know is that the more opportunities there are for the virus to spread and transmit and produce cases worldwide, the more likely it is to come across this winning formulation, a combination of mutations that allows it to make the next big jump. And it took about a year for that first big jump to be notable globally with the variants that I mentioned and several others. But we don't know how long it will take for the virus to make the next big jump. So the best mitigating strategy here is to make sure cases are suppressed globally through whatever means are necessary, a combination of MPIs and vaccines most likely. And of course, India is the prime example of that right now. Surging cases, surging deaths, and a new variant called B.1.617.2. Work is furiously being undertaken at the moment to understand whether this is yet more transmissible than the UK Kent variant, but at the moment we don't know, but it certainly does seem to be quite transmissible, and it in the UK is making up a greater proportion of cases. However, that should be said that is on the background of quite a significant reduction in cases in B.1.1.7. So we will see how that shakes out over the next few weeks, but we need to be mindful of the fact we're moving into a much more relaxed situation in terms of lockdowns, hoping that the vaccines will pick up the slack. And so it's going to be really critical to monitor that in the UK, but it's going to be absolutely essential to monitor the progress of variants like this globally, and critically to act on the findings that we see. So I think that's the end of my talk. I've tried to acknowledge key members of my lab as we've gone through, but there are hundreds of people involved in this work, and they're listed on the COG Consortium website, and I'd encourage you to check out that website for more information about the projects. Thank you very much for listening, and I hope we'll have a great question session now. Bye for now.
Right. Well, I'll try and pull up the questions. Clearly, some of the questions that started coming in were very much in line with the final part of your talk about the Indian variant. And somebody asked, how much concern is there about this variant? But particularly, what do you think the impact of this one, this Indian variant, will be on the UK as we reopen? I would say, is this a conjunction of things you really wouldn't have wanted to happen?
Yeah, it's not ideal timing, is it? By any means. It's a really hot topic at the moment. There's a lot of conversations going on right now about exactly what the Indian variant is going to mean for reopening. I guess I don't want to tackle that question head on, because I think that's as much a political question as anything else. But
absolutely, yes.
I think the Indian variant has certain aspects that are concerning, and actually, for me, the most concerning aspect of the Indian variant is what we've seen happening in India. We clearly have a exponential growth in India, and the scenes have obviously been, shocking there. It's really hard to know, particularly from remote, and this goes back to my original point about genomic surveillance being kind of uneven in different countries. It's really hard to know how much of that surge in India is driven by variant biology and how much is driven by epidemiology. And most likely it's going to be a combination, but it's not clear in what proportion. We do know, we can get a sense of what was going on in India, obviously from the data that's posted up from India, but also from our imports. We really did import quite a large number of cases, again, a kind of recurring theme over the period of over April period, particularly before we put India onto the red list for travel. And we could see from that data that that B1617 lineage became more frequent over that time period. So we actually saw 3 variants circulating in India: the UK variant B117, a second variant called B1617.1. Sorry about the names, I don't make them up. But that one, interestingly, had a antigenically interesting mutation at 484. And then B1617.2, which did seem to dominate quite quickly in terms of imports. And so we can, and that's consistent with India's data. So we could probably conclude that that variant, for whatever reason, is outcompeting the other variants, including the UK variant. And that's why PHE were quite confident in saying that that new India variant is at least as transmissible as the Kent variant, and potentially more. It's a bit harder then to know really how much more, if at all, because it's somewhat confounded at the moment by epidemiological factors. For example, most of the cases in the UK are in particular parts of the country, in a few regions of the country, notably Bolton, parts of Northwest, around Bedford, and so it's not something that's at the moment taking off and doing really well nationally. It's quite localized, and if it's like the Kent variant, we might see what happened in November, December last year again, and we'll see that outcompetes wherever it ends up. That variant outcompetes, in which case we'll be confident in saying that it's definitely more transmissible. Now, I guess the thing that's changed since last year is the vaccination program, and having a very large number of vaccinated individuals is going to be is great news, clearly, and we anticipate that the vaccine will have an effect on reducing transmission across generally, but also and critically in reducing the number of severely affected patients and hospitalizations and deaths. I don't think at the moment there's good evidence, although it was in the Financial Times yesterday. I don't think there's good evidence that there's much in the way of immune escape associated with the India variant B1617.2. The mutations are not suggestive of that compared to that other India variant B617.1, which is not doing well in the UK. I don't think that there's good suggestion of immune escape, and so I think on that basis, I am still moderately optimistic about how things are going to go, but that could be a disastrous statement in retrospect in a few weeks' time. So unfortunately, I think there's not a great deal we can do except watch and wait. It does raise interesting questions about, do we want to, for example, pursue new types of strategies? Now we have vaccines, for example, ring vaccination, targeted vaccination programs, quite an interesting idea. And obviously in the news, people have started to speculate about local lockdowns and things like that. I don't know really what's going to happen from a policy point of view, but there's clearly enough evidence there to keep a very close eye on it.
Very interesting. Thanks very much, Nick, about that. And it's almost the second question that we had. Maybe a very brief answer, even because you might have, it grows out of that. And it's, do you think there will be another wave in the UK this autumn with a possible combination of these spike protein mutations? The hope, from what you said, the hope is that the vaccine will continue to keep that under control, and hopefully not. But what do you think?
Well, I think this is not a genomics question in a sense, but the modelling around a third or a fourth wave, depending how you think about it, has always been suggested that there will be a further wave. But I think the debate is about, firstly, the size of that wave. Cases are just slightly on the uptick again after a long period of descent. We're obviously I think it's likely that these changes will happen on the 17th, and that will obviously increase the chances for more spread. So I think there will be another wave, but whether that is a large number of cases or actually a relatively sort of shallow-looking wave, I hope it won't be a huge number of cases. I think even more important is obviously how well that vaccine is going to protect against severe disease, hospitalizations, and deaths. And I think everyone's feeling confident that we're going to have that will have a big impact. But yeah, I don't think we would. I personally, I would bet on there being an increase in numbers of cases. I just hope that that will not turn into a big exponential growth like we saw last year.
Thank you. You mentioned the vaccines in there. Of course, we're being advised, promised, and so on about booster vaccine doses for the autumn in around there. Are they going to be able to take is the design of those vaccines, anything you know about that that would take account of the changes that we're seeing in the virus?
Yeah, I mean, clearly, it's going to be sensible to incorporate newly fixed mutations in the virus, or mutations that are known experimentally to change, for example, susceptibility to neutralizing antibodies. It makes perfect sense to incorporate those mutations into future vaccines. I don't really know the details of the vaccine design process, but my understanding is that technically speaking, it's very feasible to either design against, if you like, version 2 of SARS-CoV-2 in the new vaccines. Certainly technically feasible. It should be technically feasible even to design against multiple versions of the virus, and I would have thought that would be something that would be a sensible thing to do based on these kind of canonical mutations that are cropping up again and again in a convergent way. It would make sense to design against multiple versions of the virus. And my understanding of most of vaccine updates is that that's not a technical issue. It's all mainly down to regulatory issues and issues around vaccine trials. And at what point do you can you just skip through that bit like flu and just go straight to the new version, or do you have to go back and retest for safety, etc.? I would hope not. I would hope that we would be able to just update the vaccines. I'm sure that all the major manufacturers of vaccines are thinking carefully about what they will do, and I'm sure that they will update it over time. In a way, the very, very quick pace of vaccine production is sort of almost it's not a flaw, but it's almost unfortunate in the sense that we designed against a version of the virus that was a new spillover into human population, and then it's gone through a period of fairly rapid adaptation. And so in a sense, we're designing against that Wuhan strain. We're effectively designing against a bat virus, and now we have a humanized virus. So it makes sense for us to design against the human version of the virus, and that will presumably make the vaccines work better.
Again, and I like the point about hopefully not having to go through the major trials again, because we've got over that hurdle with flu, and I don't see why this should be any different to that sort of year-on-year change. Somebody with again a question this time not about the vaccines, but about incorporating the new sequences in the diagnostic tests for the initial, the frontline diagnostic tests for COVID-19. Have the sequences there been updated for diagnostic purposes, so the mutations such as Kent or India are now incorporated in that?
Yeah, I mean, there's two kind of parts to the answer to this question. Firstly is that I mentioned that the UK certainly in community testing in Pillar 2 uses very often uses the thermo-TaqPath assay, which is a 3-gene PCR assay, and probably Alan McNally talked about it in a previous talk. And of course, something that was fortuitous and unexpected was, because it's a 3-gene assay, there's some redundancy there, and it just by chance, the Kent variant, one of those deletions that I mentioned in the spike protein, causes S gene target failure, i.e. the S channel drops out of the qPCR, but you still get 2 other channels coming positive. And whilst that was initially regarded as a great flaw in the assay, it was actually quite quickly realized it was a great asset, because with the best will in the world, we can't integrate sequences much more quickly than with about a week delay, typically because samples have to go from a diagnostic lab to a sequencing lab and be processed. And yeah, you know, about a week is about right for that. And so actually leveraging that S gene target failure gave us a look into the future of about a week to 10 days as to what was going on with the Kent variant. It was a very useful thing to do. And actually it's happened again because the India variant doesn't exhibit that phenomenon, doesn't have that same deletion in the S gene, and therefore we now see a flip back to S gene positive samples. We see the inverse of what we saw last year, and so we're actually ahead of the India variant again the same way. So and that's pretty useful because actually 2 weeks ago S gene positivity was running at about 25%, and the week after it's about 40%. And so we can see that India variant is growing. We can assume that that's going to be from the India variant, even though we don't have the sequences yet, and we can model that epidemic curve even more accurately. So it's a useful thing to do. The second answer to your question is that many of the audience will be aware of these so-called reflex assays, TACMAN usually type assays, to look for the presence or absence of particular single nucleotide polymorphisms that are associated with particular lineages. Those are not foolproof because you're only assaying a couple of positions in the genome, but there are now commonly there is used a TACMAN assay. It looks for mutations like 484K, N501Y, 417, for example, and that gives a pretty good discriminatory assay for the major kind of variants of concern, particularly, you know, for example, the P1 Brazil variant that I mentioned and the B1351 South Africa variant. The flaw with these, of course, is that they take some time to set up, to validate, and to get into production. So we don't have one for the India variant yet, although we can use the S gene, luckily for that one. So they work quite well, but they maybe are not kind of fast enough to really keep on top of what is quite a dynamic situation.
Thank you. Now we're getting close to the end. A couple of quick ones here. I had one. You joked about the origins of the Kent variant. As to what do you really think it was? Was it a mutation that arose in that area? Was it imported? For not the result of a raid?
I'm pretty sure. I'm 90% sure that it arose in southeast England. And 99% sure it arose in the UK. We've never seen anything in the tree that would point to an origin in another country. So had it arose and become more transmissible anywhere else, we would expect by now to see in the phylogenetic tree evidence of that. And we don't. So there's no real reason to expect that it was imported, because all of the cases through September, October, November, when it was present, but at very small numbers, are UK cases. So there's no evidence that it came from anywhere else. Where it emerged in the UK is quite interesting in the sense that our best hypothesis at the moment is that it most likely arose in an individual, and there is quite good evidence now from the literature that certain patients with, for example, immunodeficiency disorders or acquired or immunodeficiencies are susceptible to a persistent chronic COVID infection. And some case studies, longitudinal case studies, sequencing genomes from an individual patient have shown this phenomenon of an accelerated evolutionary rate within individual patients. So our best guess is that unexplained long branch of mutations that happened where we don't have any intermediate forms may have arisen in an individual with a kind of chronic infection.
Thank you very much, and thanks. Actually, that was I didn't know whether there was going to be a quick answer or not, but that was a very good response and information I don't think many of us had before. looking at my watch, which tells me we're 2 minutes from the end of this, so I think it comes down to me now to thank you for this last talk. Thank Jonathan for the previous one. An excellent afternoon's activity. Again, thanks to all the BSMT people who've put this together. There were maybe a couple of questions still in the list that we haven't got to, which I'm sure you'll be happy if we just email them to you and ask perhaps for a quick response in that way. It would only be a couple. I'll leave that to the organisers. And for everybody who's tuned in, thank you for joining us this afternoon. I hope you've appreciated and enjoyed, been stimulated as much by it as I have. Do remember that to log in the same way for the final session tomorrow afternoon. But of course, as with all the others, they can be picked up online from a few hours after this, sometime later on this evening. The recording will be posted. So I think that's it. Thanks to everybody concerned.